Hello.
Hello, everyone. Welcome back. My name is Natalia Gorina, and I did my PhD work in the Brain Lab, and now I am a postdoc/project manager there. I hope that the ones who have joined us from early morning are still here, and those who have just arrived, so welcome. During this morning, we've heard a lot of very interesting talks, and now we have reached the keynote talk given by Sonia Koch. And I'm quite sure that most of you know who she is, and many of you are citing her research. But for those who don't, I'm going to give you a brief introduction. So Sonia is a cognitive, affective, and translational neuroscientist who investigates the role of prediction in multimodal domains in both healthy and clinical populations. She holds a chair in translational cognitive neuroscience at Maastricht University in the Netherlands, and heads the section of neuropsychology, and has multiple honorary positions and professorships around the world, such as in Manchester and Glasgow universities, Montreal, or Georgetown University. She also works for multiple funding agencies in Europe, including the ERC, and is a senior editor for leading journals in the field of cognitive neuroscience. Sonia, we're really glad you accepted our invitation for this symposium and to be our keynote talk. I think that it's an amazing pleasure to have you here. And without further to say, I think that our time will be better invested if I let you speak. So whenever you want, the stage is yours. Just let me know and I will change your slides.
Okay, thanks so much for this very kind introduction, and my congratulations to the organizers for this really fantastic meeting. It's been inspiring to hear all these super interesting results. So I hope I can contribute a little bit today with more of a synopsis of the work that's been going on in my lab for the last 10 to 15 years by now. And what I will try to elucidate today is more the neural circuitry that may underlie some of the phenomenon that we have heard about this morning. We talked about prediction, we talked about canceling hypotheses, prediction hypotheses, and so forth. So since we had some technical issues, I will have a little bit of support moving the slides, and we can go to the next slide. So there are principally three core questions that I would like to discuss with you today. So one of the big questions that, of course, we all somehow address is how we perceive and integrate dynamic multimodal stimuli and how we bind them. So we heard about motor auditory binding, visual auditory binding, I will talk a bit about. But ultimately, my bigger question is, how does this relate to more complex types of processes like speech and music? And again, we have talked a lot about why prediction may actually elucidate these processes, and I will zoom into that. Now, one question that we can, of course, address in this context as well is whether there are particular neural correlates or circuitries even that contribute to these processes. And that is what I will be focusing on today, because what I'm after is to look into more causal kinds of evidence at the brain level that will support these kinds of hypotheses. So let's move on to the next slide, please. Yeah. And this is a rather full slide, so bear with me, but I think it is actually an important slide because it may subsume some of the points that have been raised this morning. And the basic question is, do we have a brain system or even multiple brain systems that allow us to explain what we want to call, on the one hand, a forward model, or a mental model that will allow us to understand how predictions materialize at the brain level? And so the forward model is a theoretical model. It comes from the sensory motor domain, or rather the somatosensory domain, as we've heard earlier on. And what it basically tells us is that we are not just executing a motor command, but we also have what we call an efference copy. So in a sense, simply a copy that will allow us ultimately to compare what is the actual outcome of an action relative to the prediction of an action, because we have an expectation about what this action should look like. And so this ultimately allows us to compare actual versus expected sensory consequences of an action. And interestingly enough, in this context, there have been ideas on the anatomical correlates. And one idea is that we're not just looking at cortical brain structures that allow us to make that active comparison, but it's actually a whole pathway called the Cortico-cerebellar pathway, in which the cerebellum plays a critical role in allowing us to compare and potentially trigger also an error signal to the cortex about what is expected and what we actually experience as a sensory consequence of an action. Now, there have been many ideas about this, but what is the most critical thing here is that we actually have a neuroanatomical conceptualization of what a prediction could stand for. So not just a theoretical concept. And what is interesting is that in more recent years, a lot of people have actually considered whether this is a model that can enlighten us not only about the sensory consequences of motor actions, but potentially about sensory consequences of any form of plan that we want to execute. That goes as far as going towards cognition. I'm not going to push that far today. I'm going to stick to perception. But the question is, indeed, can we generalize this type of prediction model that has neuroanatomical correlates to something more general than just motor action? Okay, next slide, please. Of course, we have our tools, and I want to quickly let you know how I'm going to work and with what I'm going to work. On the one hand, we have these indices in electrophysiology, which we call suppression effects. We have been looking specifically at early event-related brain potential components, such as the P50 and the N100, which can be source localized in the secondary auditory cortices. And what we do know, and you see that here on the right-hand side in this exemplar example, is that externally produced sounds lead to a larger N1 amplitude than self-initiated sounds. So in the context of a motor sensory binding, this is what we tend to see, and this is considered to be a suppression effect. And now, interestingly enough, we see similar kinds of suppression effects also in this time window in the multisensory domain. And we also have shown that suppression, when a stimulus in the auditory modality is temporally predictable, leads to similar kind of suppression. So we already have indices in the literature and based on different kinds of binding mechanisms that this suppression effect tells us something about facilitated processing. And as Sophie already mentioned beforehand, there is something going on before the target sound is actually perceived, namely the alpha-beta suppression that seems to be a marker of immediate stimulus occurrence, and I will show a little bit of data in that context as well. Now, the big question that we're facing at the moment in the literature is that there are people who probably go along with the idea that the cortico-cerebellar pathway is quite critical in regulating the comparison between actual and expected sensory consequences. And And on the other hand, we have very strong proponents of an exclusively cortico-cortical regulation. That means top-down regulation of sensory cortices. But as I already asked Emily this morning, maybe it's not one or the other, but maybe it's about looking at the time course of these effects, namely looking at anticipation before the sound onset, for example, and the evolution of the consequences of a prediction over time that may actually help us to understand this better. Unfortunately, what I can't show in this graphic display up there is a phenomenon that we very, very often see in combination. That is, we see early suppression effects and later on enhancement effects of this facilitation that we talk about. So maybe it's one story after all, but I'm only going to talk about the suppression effect today, but I want to invite that type of thinking and discussion. Okay, let's move on. So just to summarize, what we're going to look at is at a generalization of the forward model. The flow of information from a motor command to the cerebellum back via the thalamus to sensory cortices leading to suppression. Let's move on, please. Now, of course, nowadays we have much more fancy tools, including TMS. But when I started out this work, I actually still work a lot with patients. And so one interesting way of looking into a causal relationship is to look at lesion sites. And this is exactly what we're doing here. So in the following two experiments that I will show data on, I worked with patients who have cerebellar lesions, and what you see at the top of this slide is a lesion overlay. And interestingly enough, what you see right away is that the lesions were all over the place. As a matter of fact, that should critically work against us because it's not a specific location in the cerebellum that was damaged in these patients. And if there is anything about generalizing the cerebellum's contribution to this prediction comparison, then we should see an effect there. So one critical hypothesis is, do we see any form of alteration in these patients when we look at self-initiated sound? Because they're lacking the capacity to actually make that critical comparison and elicit an error signal to the cortex. So let's move on, please. I quickly want to introduce you to the paradigm that we're using. This was actually work done together with my dear friend Erich Schröger in Leipzig. It's called the N1 suppression paradigm, and a lot of the Spanish collaborators know about this work quite well. So we're actually using a very basic paradigm where you press a button and immediately with the button press, you hear a sound. We record the sounds and present them in a exactly similar timing as the sound presentation in the auditory motor condition. And we use in C, a motor-only control condition to try to minimize by subtracting this condition, so C from A, the contribution of the motor activity per se. And if we move on to the right-hand side of the slide, what we see is in blue, the auditory-only response. So we're looking at the physically identical sounds compared to the red auditory motor corrected response. And what we see very nicely in the control participants is a massive suppression effect when the sound is the consequence of a self-induced action. Now, as I said before, the critical question was, do patients who have cerebellar damage show a similar kind of suppression effect? And as a matter of fact, what we see is that they don't. So first, evidence that there may be something going on in terms of this comparison mechanism that is apparent if you have a lesion in the cerebellum. Next slide, please. Now, one question that we can ask ourselves is, if we block these conditions, so A, B, C, is that sort of a self-fulfilling prophecy? So in a next study, and in many ways a replication study of the first results, we compared directly the mixing of the conditions, which you see on the right-hand panel, and the single presentation of the conditions on the left. And maybe we can just move on one click. So if you look now at the controls in both the single and the mixed condition, we see again a replication of the suppression effect in the N1. So again, the blue on top of the red line. And interestingly enough, and nice enough for us, we replicate our prior findings in the block condition, no suppression effect for the cerebellar patients. So that was quite promising for us to see a second time. All right, let's move on to the next slide, please. Of course, we are interested to also understand whether this doesn't just work for sounds, but maybe also for more complex kind of motor actions, such as voice production. This is particularly interesting in the context of research that we're doing in schizophrenia, where we have the situation that patients may suffer from auditory verbal hallucinations. And one idea is that they actually lack this, not just the binding between the motor, so the sound production and the feedback, but actually that the comparison mechanism is at fault. And what we did here is not only manipulate the expectancy of the sound, which was 100% if you press the button and the sound is elicited, or in this case, the voice, but we altered also the probability of occurrence that the feedback will be your own voice relative to not. And what we see is one of two things. If we go with 100% probability that the button press will lead to the elicitation of your voice, you again see the same kind of suppression effect, even for these complex stimuli, such as voices. But if you alter the probability, what you end up seeing in the N1 range is a change in terms of the amplitude suppression. So the less likely the voice will occur as a function of your own action, the less suppression you will see, as you see that the red line approaches the blue line on the right-hand side. Okay, so let me get to a brief summary, interim summary on the next slide. So what we can show with this type of causal approach is that the cerebellum modulates the auditory N100 suppression effect independent of the allocation of attention and selectively. I didn't talk about that, but also independent of the temporal proximity of external sounds, which we manipulated in another study. The N1 suppression also works for voice production, which allows us to actually control for a lot of motor artifacts if we look at natural production. And as I just said, this has implications for psychiatric disorders, such as hearing voices. And for those of you who are interested, we just put out a concept paper recently that can explain that a little bit further to you. All right, let's move on to the next slide. So I said before, we're interested in understanding whether this neurocircuitry can be generalized to other binding phenomenon. But before we go there, so we have had causal evidence from the cerebellar patient data. I would like to get into quickly into a data set that allowed us to actually look into the structural foundations of connectivity between the cerebellum and the temporal cortex, which is critical when we look at motor auditory binding. Next slide, please. So I'm not going to dwell a lot on all the different tests that we did, but the approach again was a patient approach. And in this particular context, we looked into what we call lesion symptom mapping in patients who had strokes in the posterior superior temporal sulcus of the left hemisphere. Now, this means these patients, and you see a sort of the lesion overlay on the right-hand side, top picture. This means these patients had spared Heschel's gyri, meaning their hearing was good. That was not damaged, which is quite critical in this context. So we also screened for that. But we did then a couple of what we call temporal discrimination tests that allowed us to test, on the one hand, understanding micro-patterning of time sensitivity, as much as then going into what we call phoneme discrimination, pseudo-word discrimination, and word discrimination in these patients. Again, going from lower level temporal coding to higher level temporal coding in speech. Now, this is interesting on two accounts, because this area in the left temporal cortex is denoted as sensitive to particular timescale coding of information, so fast transitions, as we have heard about in what we call asymmetric sampling. And so what we see here is that patients who have in the lesion overlay now a maximal seed region, which you see on the right-hand side bottom figure, patients who have trouble with threshold discrimination, phoneme discrimination, pseudo-word discrimination, word discrimination, seem to have a maximal lesion overlay in this particular posterior temporal cortex spot. So this seems to be an area that is at least partially related to this fine-grained temporal discrimination encoded in these different stimuli. Now, what we then do, can I have the next slide, please, is take that seed-based region, and we start tracking in healthy age-matched controls the pathways that are actually linking with this particular seed region. And on the left-hand side, in panel A and panel B, you see very nicely that this is a region that links with the dorsal and ventral pathway. So this is all as expected, and that's very nice that we can actually confirm that even based on lesion symptom mapping. But what we were particularly interested in was whether we see a mirror image in connectivity between this left hemisphere temporal cortex region and the right cerebellum. And this is what you see traced in red. Can we go to the next slide, please? And just to show you, nope, that's too fast, thank you. So what we see on the right-hand panel is now the tracing from this seed region in the temporal cortex in red that traces into the cerebellum. But critical for us, we also tracked from the same region in the cerebellum now into the primary motor cortex, so going frontopontine rather than temporo-pontine. And what we can clearly see is that along the path, we really have a split of fiber trajectories that go temporal relative to motor frontal. So we think with these types of data, we can demonstrate that there is not just a functional reality, but there's also a structural reality for this kind of connectivity that may be supporting the network that I introduced to you before. All right, so let's move on to the next slide because I want to summarize again. So we seem to be able to show that there is structural connectivity between the temporal cortex and the cerebellum that supports the extraction and representation of the temporal structure of simple and complex auditory signals, as we have shown through the lesion symptom mapping. All right, so we have established some functional causal evidence with the cerebellar patients. We have established, hopefully, some structural evidence through the lesion symptom mapping in the cortical patients. Let's move on to the next slide, because if we really want to get to a generalization of this forward model, in a sense to a mental model, we need to test whether there is indeed generalizability. And we have done this in the context of sensory prediction, so multisensory stimuli. So let's have a look at that. Maybe we just unfold the slide here. Yeah, yeah, it's fine. It's fine. So rather than working with a motor-induced sound, we worked here with visual stimuli and auditory stimuli that we bound in time, either in a predictable fashion, which you see in panel A, so the binding window was always the same, or we had a random binding of the visual and auditory stimuli. So they could either be very closely related in time, they could be related exactly the same way in time as in the fixed auditory condition, or there was no binding at all. So the critical question was, do we find suppression effects? And as you see, again, on the right-hand side, we're zooming into the N1 component in blue for the unpredictable, that is condition B, and in red for the predictable, condition A. And what we do see is, again, a suppression effect in the N1. As a matter of fact, we see something even earlier in the P2 range, in the P1 range, I'm sorry. But we do see that this effect is far less enhanced as it was for the motor auditory binding. But what was really, really exciting to see is when we now test again with the cerebellar patients, exactly the same sample as before, we see no suppression effect, as we've not seen a suppression effect for the motor auditory binding. All right, so let's move on to the next slide, please. I don't want to just talk about patients, even though for me they're really, really a good example. What I would like to share with you is some fMRI functional evidence that we used in the context of multisensory emotional and neutral stimuli, where we had three conditions, as you see on the left-hand side of the slide. In condition A, we had a stimulus that was just an auditory stimulus with visual stimulus staying fixated and still. We did control for this in pilot studies also without direct stimulation, and the results are the same. We had a visual-only condition, and the visual-only condition is now a dynamic visual evolution of a body movement that goes along with the vocalization in the C condition, as you see here exemplified in the static picture. What I would like to simply deliver as a take-home message here was what we call a conjunction analysis result, which you now see on the right-hand side. So we try to establish that there is a structural relationship between the temporal cortex and the cerebellum. And what we actually see in this conjunction analysis, where you ultimately see enhanced activation for audiovisual compared to visual, and that in conjunction with audiovisual larger than auditory stimulation only, you see the network that I was so interested in, and that is the bilateral superior temporal gyrus activated in the top left panel, the bilateral mediate geniculate body of the thalamus on the top right panel, and you see bilateral activation in the cerebellum. So even with much more complex speech and dynamic visual stimulation, we see this network in action, which is, I think, a nice piece of evidence. So let's move on to the next slide. So just to show you that we also tested this very same paradigm in EEG. What I simply want you to take home from this slide is that we see on the top left, again, a very nice N1 suppression effect, where for the audiovisual stimuli in red, you see suppression relative to auditory only. So this goes along with the idea that very likely the visual information has very high predictive value for the auditory sound quality. And on the right-hand side, we now look at the stimulation time window before the target onset, so where we now can compare actually the time frequency domain, and you see that the visual alpha / beta, because that's the lower border of lower beta already, is very comparable to the audiovisual, while we don't see such a suppression effect for the auditory only condition. Just to show you, if we do one more click, please. Hello? Okay. We also see a suppression effect if we look at the stimulus quality. So plotting across all modalities, anger, fear versus neutral. Interestingly enough, the quality of the stimulus per se also leads to a suppression effect. However, what we did not find in this investigation is an interaction between the modalities and the emotionality of the stimuli. We think a lot of it has to do with the timing, but I don't have time to look into this with you at this particular point in time. If you're interested, we published on this in a Frontiers paper together with Sarah Jessen, a collaborator in Lübeck. All right, so that gets me, I think, to my summary and conclusion slide. So what I've been trying to entertain with you is that we have some neurofunctional reality for prediction. And it is interesting to start out from the internal model idea, where we see that this really serves to better understand motor sensory predictions as much as multisensory predictions. I hope I could convince you somehow that a re-entry system such as the cerebellar loop may actually contribute to successfully predict the sensory feedback and consequently suppression in the temporal cortex. and that the separation of slower and faster dynamics in speech processing and dedicated temporal processing does indeed relate to the cerebellum, as shown in the data with the lesion symptom mapping. So all in all, I think it's an interesting combination of results. It may, in many ways, if I go back to Emily Thompson's talk this morning, go more along with a cancellation idea. But as I said, what we need to actually really look at is at the temporal unfolding of whether it's just suppression or also enhancement, under which task constraints these suppression and enhancement effects occur. And there's clearly quite a bit of work to be done in terms of that. All right, that brings me to the end of my presentation. Of course, I would like to thank a few people and funding agencies. This was work done with Annika Stockert, who was involved in the lesion symptom mapping data, Sarah Jessen, the multisensory work, Franziska Knolle, Michael Schwarze, for the motor sensory suppression work, and Anna Pinero, who is guiding all our work on suppression effects in schizophrenia. Thank you very much for your attention, and I'm happy to answer questions.
Thank you very, very much, Sonia, for this super clear talk. I think that there are some questions right now on the question. So if you hear me, I will ask you the first question, which says that the absence of suppression effects in cerebellar patients seem to go in line with Constantina's finding of enhanced connectivity between sensory areas and cerebellum with larger perceptual attenuation effects. Do you think that self-generation studies should look further into the contributions of cerebellum in the sensory attenuation effect?
And I could not agree more. I was actually very thrilled to see these results. And for me, the particularly interesting aspect is now to actually really look whether we can compare attenuation and gating. So I'm quite curious to think about this more. In particular, if you think about the situation with schizophrenia or even first incident psychosis, where people tend to think a sound comes from outside, so external, even though it's actually potentially their own aberrant attenuation, is an interesting case scenario to test this more closely. And potentially even with the types of paradigms that Constantina has presented. So I'm quite curious. Yeah, but I go along with that, totally.
Okay, then we also have another question that says, what do you think might be the contribution of direct cortico-cortical, so non-motor, for example, non-motor, motor auditory connections in comparison to the loop via the cerebellum?
There is no doubt that we can think about prediction in the sense of a cortical representation here. I think it's just that, for my taste, things would have to come together. At the moment, the cortical models will only talk about predictions from one cortical area to another cortical area. And what I'm trying to argue is, in what way can we measure the contribution of error feedback? And I think we need to either understand whether that is doable entirely at the cortical level, or whether these error monitoring areas of the brain, of which we don't just have the cerebellum, but we also have the basal ganglia system, contribute critically to the fact that we can really adapt to kind of unexpected events. So they may not just be erroneous, but they may just be unexpected. So there's a gradient in what I want to call a full error response to something that is unexpected. So I think there's quite a few questions open that we need to address, and we're working on this, and I hope people join.
Thank you. Okay, so now they are saying like, thank you for the great talk. We have also seen some enhancements in some of our results. Do you think these enhancements have to do with the building up of predictions / forward models?
For me, what is still sort of the elephant in the room is how we test on the one hand, and how much attention plays a role on the other hand. So if I go from a classical event-related potential model, we still have the idea that very early components may be almost processed without attention, even though I don't believe that. And the more later components may be a lot more attention-driven. So what I clearly see in a lot of our data, if we open the time window to later time windows, we see this flip between suppression and enhancement. So we need to understand at what point in time enhancement is actually driven by really attentional boosting of a prediction, yes or no, and whether what we see early on is sort of a first, okay, this is expected, this is not so expected, this is actually really faulty. So I don't want to just go into this automatic controlled kind of dichotomy, not at all. But I think we need to understand the timing of these effects better as a function of the task and how much attention is given to the properties that we're looking for in a task, until we can resolve that question fully.
Great, thank you. And I think that we can go for another last question. Thank you for the great keynote talk. In evolutionary research, the cerebellum is sometimes highlighted because it has supposedly expanded a lot in size during recent primate evolution. I was wondering if you have an opinion on the role of cerebellum in language evolution.
Yeah, as a matter of fact, we have written about this ourselves in a TICS paper already in 2010, because we were speculating about the expansion of the cerebellum and its effect on the evolution, in particular of the frontal cortex. And it seems that there is a really, really strong correlation, and that it almost appears as if the cerebellum hadn't evolved, our frontal cortex wouldn't have evolved. And of course, that has massive consequences for any kind of higher level cognition, whether it be language or any other form of complex cognitive procedure. It affects our learning, it affects our selection, it affects our control mechanisms. So I think, yes, there's a lot to be said about that, and I would definitely sign up on that.
Thank you very much. So again, thank you for this amazing keynote talk. I think that it's good for questions right now, and we are going to give, like, let's say a couple of minutes until we go to session 4.
Yes.
And we start with the roundtable to discuss a little bit about the gender issues that happened in this world.
Yes.
Yeah, thanks again for the opportunity.
So you can go to the session number 4 by clicking schedule on the top left of the screen, and let's meet there in 3-4 minutes, let's say. Okay?
Thank you.
See you soon.